All right, so in this experiment, all right, we're going to be looking at the effect of light on algal cell growth. You have a little quick guide that kind of looks a little bit like this that you'll be using as a reference. You have detailed instructions specifically on what to do. And so now I'm going to show you how to set up this experiment. So what you'll need, you need your test tube rack, you need 3 of these 15 milliliter tubes, and then you should have picked up an algal culture when you picked up your kit, right, separately, that has live algal cells in it. And so what we're gonna do is, I'm just gonna grab this. I recommend you use spring water. So from a bottle, not your tap water. So tap water varies. Some of it's chlorinated. It might affect the experiment depending on where you're getting it from. So I would say just get a regular bottle of water that you would drink, and that's what you'll use to set up some of these experiments. That'll be the safest thing for the cells in the experiments. So what you're gonna do is you're gonna take this algal culture first and then dilute it into these 3 tubes. So you have enough in here to put 1 milliliter in each of your tubes. So what I'm gonna do is I'll take the caps off. 1, 2, 3. Now you'll use your pipette, okay, and open this up here. So the pipette should be marked to the 1 mil mark. You go find it. It is around here. I recommend getting a marker. I didn't bring one in here with me right now, but there's a little line here. I would mark it with a Sharpie so that you can easily find it and go to the same mark multiple times. Otherwise, you're just going to be guessing, and it's not going to be good. So that's 1, 2, 3. Okay, and you should have some left over. So in case you need to redo the experiment, you mess up something, you might be able to set it up again. You're just going to bring this up to the 5 milliliter mark. Okay, so just a 5 milliliter on each of these. And this one too. It's important that you put the same amount of algal culture in each tube, and you put the same amount of water in each tube, because the idea is you're going to be counting the cells in these cultures over time. And if you start off with different amounts, obviously you're going to end up with different amounts in the end. The idea is that you start off with the same amount in every tube from the beginning. So I'm putting the caps on. If there's a lot of air still in here, you may want to, you can mix them a little bit like this. Make sure the cap is tight when you do that. You may want to open the cap up a little bit, but if you let the cap become too loose or take the cap off, there's going to be evaporation and it's going to change the experiment. So I recommend not doing that. Keep the caps on, but maybe not totally tight, just a little bit loose, and then that should be fine. Now, the 3 different treatments that you're going to be setting up here, one is going to be total darkness. Okay, so what you're going to do is take your aluminum foil, wrap it around the total darkness tube, tear this off, seal it, put it down. Okay, so it's going to stay in total darkness the whole time of this experiment. There's a second one. This one you're going to be removing. Okay, so it's going to experience light during some time of the day, and then other times of the day it's going to be in darkness. So you're going to kind of have to make a note for yourself to switch this off and on, or else it's going to mess up the experiment. So it's like a 12 on, 12 off light cycle. And then this one is just going to stay uncovered. It's going to get 12 hours of light. That means you need a light source. You're going to need something like a little desk lamp, something that you could plug in. This is an LED one, so it's not going to take up much power as I use it. And then when you turn this on, I would angle it, in a way, obviously, at the cells, so it's going to hit them. Now that's the basic setup, but there's one thing still missing, is that you need a start concentration. So that means right now you need to know how many cells are there per milliliter, or else you won't have anything to compare it to. So you have a starting concentration. You don't have to measure all of these, you just need one of them. So essentially from one of these tubes, probably just this one, because it's the easiest right now, what you would do is get your counting slide, make sure it's cleaned and dried from the last time you had used it, get your little microscope, and get you that culture, right? You'll take a small sample out of the culture. All you really need is just enough to add to your slide, just really one drop, is really all you need. It would be a good idea if you had mixed it beforehand, just in case, inside the water. So I would mix it, then take your sample. Then you're going to load that sample into the chamber, and once it draws across, you know it's in there, and that's pretty much it. Then you'll essentially take that sample, put it onto your little microscope stage that you have, get your microscope, the light on, go ahead and set it in place, and then you're ready to look at it. You might find that you have a way of using the microscope that you prefer. So right now I turned it sort of kind of sideways. A lot of people might use it this way, but you might find maneuvering it that way is difficult for you. There is not one correct way to do the microscope part of it. It's whatever works for you. If whatever you find works, you can see the slide, you can see the grid on the slide, and you can see the cells in the grid, that's good, okay? And if you could only see it when you're looking through your phone, and you can take pictures of it or video, that's fine too. As long as you can see it, it's a good way of doing it. If you can't see it, then you need to change and do something different. So changing the angle that you look at it, that could help. Whatever you find works, right? So then you're gonna take a photo, or you could do a direct count right there, however you wanna do it, in the squares, and then I have a separate explanation of exactly how many squares to count or what size squares to use, and you're gonna take that count and record it. That's your start number. Right, so you have a starting number of cells, and then you're going to wait. All right, and then according to our plan here, you're going to then be recounting the number of cells here after 2, 4, 6, and 8 days. So this experiment is going to run for over a week. So every 2 days, you're going to come back here and count the cells. When you do that, you're going to have to count in all 3 treatments. Okay, so that means you're going to have to take cells from the first one, put it in here, do the count again, see if it changed. I think it would, but I don't know. You'll have to find that out. The 12 on, 12 off, light, dark. Maybe that changes too. Maybe it doesn't. I don't know. The dark one, maybe it stays the same. Maybe it goes down. And who knows? Maybe it goes up for some unexplained reason. That's why we're doing an experiment, right? 2 more days go by, do it again on the 4th day, 6th day, 8th day, and then we finish, and then it's done. So every other day you're gonna have to do this. However long it takes you to take these readings is gonna be how long it takes to do that work. So if it takes you a very long time and you're having a hard time with this, hopefully the practice will get you better, so that each successive time you do it, it takes less time to find it. Don't be super frustrated if the very first time you're doing it, you're finding it difficult, right? Because you may. That's totally understandable that you would, but remember, once you get the hang of it, just like anything else, it'll become easy and it'll be very quick, right? So it's going to be so hard every single time you do it. It'll be very easy after you've done it 2, 3, 4 times. And you're doing this with the other experiments in the lab too, with yeast cells. You're going to have a bunch of experience focusing with this microscope and the base, so I don't think it'll be much of a problem for you. And that's pretty much it. That's just the setup. It's very simple. Algae in the tubes, water in the tubes, set them up, all light, all dark, intermittent, light and dark, and then you're going to be taking readings. And that's pretty much it.